Okay, so we got some introduction to VCF files. Now let's turn to look how to actually work with it in Python. So we're going to use the pyvcf module, which I vilified and said some bad things about, but I still think it's like the most decent option we have. So for this example, we're going to work with a 1,000 genome project of 2,500 individuals, which you can download at this location. So I gave you the full URL. And I already downloaded it and put it in C downloads. So you see it has a gz suffix, meaning I can open it with a gzip. And for now, I just want to look at the actual text. So I just open it with gzip and read the first 100,000 characters within the file and just print it. And you can see what you have within it. Again, all the headers that we mentioned. We have quite a few of them. And now we have the names of the different columns, which you can see we have the regular ones, and then we have 2,504 IDs. So each of these is like some kind of ID that they gave to like a specific individual for every one of them. And then you start having the actual variants. And here they don't give much information, they just give the actual genotyping of every person. And you can see, basically you just get a genotyping of each person. Now, if I want to actually starting to parse and process the data, I can import the VCF module. And then I create a VCF reader. So I do VCF reader. That's like the name of the class that I give the file name. And because it has a gz suffix, I say that it's compressed. So it automatically knows how to open the gz compression. Now, first thing I can access is the metadata, which is just different parts of the header. And it basically just gives me a dictionary, which I can iterate if I want. So I can go for each key and value and see what I get. You can see it's just a basic key value pairing of different metadata. So I have the file format, which is it's VCF 4.1, which was created in 2015. It gives a link to the reference genome that they used. So they didn't exactly use the 37 version. They used some kind of modification that was specific to the 1,000 genome. And you have the URL if you want to download it. And also saying like the source, like what pipeline they used to create it. Now on top of that, we also have some info. This just gives you the metadata, like the description of the different infos. So for each info field, again, it's just a key value pairing. So I get a name of the info field and then the info object, which has a few fields. One of them is the type, which tells you what type the info field is and what the description of it. And you can see all the different kind of fields that we have. We have quite a few of them. Most of them are not that useful, I guess, but you have some quite common ones like the AC and AF, which we mentioned. You can see them here. OK, so we dealt with the headers. Now we want to actually read the data. First thing, we may want to get just the name of the samples, the names of the samples. And we see, like we saw before, we have 2,504 of them. And you have the IDs of the first 10. Again, the VCF reader samples will just give you a list of strings with the names of the different samples. Now, if you actually want to start reading variants, you can just iterate through this VCF reader using the next function that we saw many times before. So VCF reader is actually nothing more than an iterator, which gives you one line, one variant, one after the other. And that's a good thing that you did it that way. Otherwise, it would take you like maybe centuries to just open the file because it would try to parse everything. But that's not how it actually works. It just parses it on demand. It's doing this kind of lazy operation, thankfully. So every time that you iterate through this, it reads additional line, then parses it, and then gives you the result. So that's how it works. Spare both CPU time and memory. So it wouldn't be feasible also in terms of memory in many cases to just load everything to memory. Of course, if you just want one variant, you can use the next function. If you want to iterate through the entire thing, you can just create a for loop. So if you did something like for record in VCF reader, you would just iterate through all the variants one after the other. But for now, we just want a single variant. Again, it's a very big file, and pyvcf is incredibly slow. So that should be enough for us. And now the record, it gives some kind of object which has different fields. It has a chrom, pos, ref, alt, ID, all the kind of fields that we know. So you can actually see that this variant is in chromosome 17. That's what we downloaded at the very beginning of the chromosome, so at position 52. And we have a C nucleotide that could turn into potentially an A nucleotide, and it has an RSID. And now you will note that the alternative is actually a list. You can see it according to these square brackets. And also, if you check the type, you can see that it's actually a list. Now, in most cases, it will be just a single option, so you will only have one alternative. But like we saw, in some cases, you could have an alternative allele with a few options. And then you can also get the info for this specific field. So you can see the allele count, allele frequency, or the rest as a dictionary, which you can just access. You have some nice shortcuts, so you can check the type of the variant. It just automatically does it for you according to like the reference and the alternative. So in this case, both the reference and the alternative are just one nucleotide long. So pyvcf knows that it's a SNP according to this definition. And you can also check various kind of Boolean fields. So is it a SNP? Yes. Is it an indel? No. And you can also get all the alleles together. So you get the first one, the reference C, and then all the alternative ones if you want. Now, if you go to record samples, you will get, again, a list of all 2,504 individuals. But this time, it will actually give you the calling of each one of these samples with respect to this particular variant. So you can see that each of these is a call type that refers to a specific individual and has specific genotyping. And let's look at one of these as an example. So we go to record samples 0. OK, so now we look at the first variant within the VCF file. That's the record object. And within the first variant, we look at the first sample, the first genotype. That's the samples 0, 0. And we get a calling of this specific genotype, which you see here. Now, again, you can get the reference to the sample that it refers to, and just getting the name of the sample. You can get the indices of the alleles using GT alleles that you see here. Or if you want the actual bases, you can get it as bases. And then it just goes to check. It knows that 0 refers to the reference allele, which is a C in this case, like we saw here. So this particular individual has twice C. And you can ask different questions. Is it a variant? In this case, no. This person does not have this variant. Is it heterozygous? No, it's not a heterozygous. It's a homozygous in this case, and so on. You can also, if you are interested in a specific kind of sample, you can retrieve it directly using the genotype function, getting the ID of that sample, and it will give you a call of that particular sample. Now, sometimes you might be only interested in the individuals that have the variant. So you can get, for example, you want all the people that don't have this variant at all, so they're homozygous reference. You can use this function and get a very large list of most people because it's a quite rare variant. Now we print only the first 3. Or if you want the people that actually have this variant heterozygously, you can get the list of them. There are 3 of them, so we have 3 people that have 0, 1, or 1, 0, so just one copy of this variant. Or you can get the homozygous alternative, so people that have actually 2 copies of this allele. In this case, you have none. Okay, it's a rare variant, and we don't have anyone that is homozygous with respect to this variant. You can also get the unknown, so sometimes we don't have perfect calling. So some samples we have sufficient data to do calling, and in some samples we don't have calling. But in this case, you can see that, for example, the num called, if you call it, you get that all 2,504 samples were called in this case. And you can get all the numbers. So the numbers that have no variant, the number that have are heterozygous, homozygous alternative, and unknown. And yeah, that's basically it. So you can just iterate through the file and start getting the data and do whatever analysis you want. Any questions?
So in what cases would we prefer to just parse by ourselves and not use this?
Okay, so I hope you got the impression that this is a very kind of neat and nice interface, this package. So very nice interface, very nice to work with. So if it works for you and your data is not that big and you can just work with pyvcf, then go for it. It's probably the most convenient way to work with the data. If it's big and it won't scale, then I don't know a very useful Python package. Like I said, it's not a very complicated format, so you can just parse it whichever way you see fit. You can use pandas, you can use regex. Okay, more questions? But again, maybe there does exist some nice VCF package out there, which is perfect, and I'm not aware of it. If you encounter one, then let me know. But yeah. Okay, let's see a more concrete example of something we can do. Let's run the code that, again, we'll go over this VCF file, and we'll try to build a DNA substitution matrix. So we're going to build a 4x4 matrix that for each entry will give us like the percentage of this particular substitution. Okay, so let's see how we build it. Okay, so we create a list of all 4 possible bases, and we create a data frame with 4 rows and 4 columns according to these 16 combinations. And we initialize everything as 0. Now again, pyvcf, as you can see, is very slow. So we just iterate through 5,000 records, and you can see with this progress bar that even 5,000 variants is very slow. So we have millions of variants in 1,000 genomes, so you can only imagine how long it would take to parse everything. Okay, so for the sake of our sanity, we're going to limit it to 5,000 records. I create this float progress that you see here so we can visualize how it goes. And now I take this VCF reader, open it like I opened it before, and then I use the islice function from within the itertools module. Now to remind you, the islice, what it does is it simply slices an iterator. So it gives an iterator that could you could iterate through indefinitely, and it limits it to some number. So in this case, 5,000. So we want to get some iterator that will give us only the first 5,000 elements. So we use the islice, and we enumerate over it, so we can also get the index. So we can then get the index and the record of each variant. We update the progress bar. And in this case, we're only interested in SNPs, so we won't process any indels or something that is more complicated. So only if that's a SNP. We then want to go over all the alternative. I remind you that this is a list of all the alternative nucleotides. And the record info AF will give me, again, a list of the allele frequency of each of these alleles with the same order. So I can then zip them together, and for each allele, I can get both the nucleotide and the frequency of that nucleotide. And then I simply go to the matrix in the position of the reference. So that will be the row. The row will describe the reference nucleotide. And then the alternative will be used to indicate a column. And I increase it by the frequency of that variant. OK, so I keep increasing it. But finally, I will divide it. I want to normalize it by rows. So I want every row to sum up to 1, because I want it to describe some kind of probability. So if I begin with some reference nucleotide, I want to think about it as some kind of like, what's the probability of this reference nucleotide substituting it to each of these other nucleotides, given that it is substituted. So by definition, I keep the diagonal 0. So A to A is not a variant. So to do that, I simply sum up everything column-wise to get a sum of each row and then use the div function with the proper axis to divide it accordingly. So simply dividing all the elements within row with the summing up of this row. And then I get this nice matrix that you can then think about as some kind of probabilities. So you can see the probability of each kind of substitution. And I also wrote some code here that turns it into a nice heat map. I won't go over it. It's very standard, and we've seen many of those before. So you can actually see like what's common and what's less common. And I think it's very noticeable that you have this distinction that we expect between the in-group transitions. So purines like A and G are more likely to substitute between one another. And the pyrimidines like G and T, again, are more, and substituting between them, C and T is again common, and substituting between them, so transitioning between purines to pyrimidines is far less common, as you can see according to these numbers. Questions? Okay, so yeah, I think that will pretty much summarize this discussion of VCF, and it's going to show up in the upcoming exercise. So we're going to get the opportunity to practice that. Now, final topic that I wanted to go through is multivariate analysis, which is some kind of a leftover from the previous two weeks where we discussed statistics. So multivariate analysis is, in essence, just analyzing more than two variables. And I think one of the main motivations that we have to do that is what is known as the problem of confounding, which you encountered during your exercises. You had a very explicit question about confounding, and you read about it. But I will repeat it again briefly. So yeah, virtually everything that we worked with up until now, we just worked with two variables. This is called a bivariate analysis, so just two variables, and it doesn't matter. We looked at all kinds of different scenarios. So we looked at two continuous variables and two categorical variables, and one continuous and one categorical. And for each one of these combinations, we look at different kind of tests. But we didn't look at any test that actually considers more than two variables at a time. But again, like we said, due to confounding, this very often might be important, because if we only look at two variables at a time, we might end up with something that is known as spurious correlations. So as a simple example, let's say that we're interested in studying the association between alcohol consumption to mortality. So we test it, and we see that there is indeed some positive correlation. Like we said in previous lessons, you cannot really conclude causality when you just see association. I mean, thankfully in this case, there is one possibility that you can rule out. So we know that mortality doesn't affect alcohol consumption. So after people are dead, they don't drink anything. So at least we know that if there is some causal relationship, it has to go from consuming alcohol to dying, and not the other way around. But we might have this problem of confounding. So we know there are many relevant variables that could affect different individuals, which could affect both of them. For example, sex is a very important confounder almost every time that you study individuals. So we know, for example, that men tend to drink more alcohol and they tend to die younger. So this could explain some of the association that we see between people drinking more alcohol and dying younger. And you can think of all kinds of other variables like income and other factors which could affect it. And if we just study alcohol consumption and mortality, we cannot really know whether this association is due to all of those confounders. So thankfully, we can do what is called controlling or adjusting for confounders. There are different ways to do it. One option is called stratification, meaning basically to perform the analysis within homogeneous groups. So for example, if we want to account for sex, we can say, okay, let's do the analysis for men and women separately. And we can then just report the per group p-value. So we can say something, okay, it's significant in both men and females, or it's only significant for females and not for males. Or we can then somehow merge the two of them. This will actually be discussed in an upcoming bonus question that we'll make sure to circulate among you. So we can see how we can take per group p-values and then to merge them into something global. Or another way that we can work with it is using multivariate models that will consider a few variables at a time, which is what we're going to focus about today. Now, it's important to keep in mind that either way, we can only account for confounders which are both known and observed. So of course, you cannot account for something if you didn't even think about it, which is always a great problem. I mean, it's actually like virtually impossible to prove some causal relationship based on observations alone, because like some very skeptic person can always point out that there could be some kind of hidden confounder that you didn't even think about, but could nevertheless confound your results. Again, something that is important to keep in mind, but very often there is nothing we can do about it, other than trying to think really hard maybe and really make sure that we come up at least with the most plausible kind of suspects. Another limitation is that it has to be observed. So maybe there are some kinds of confounders which conceptually you can think about. I don't know, maybe something that can affect both alcohol consumption and mortality is someone's attitude towards life. Okay? So they could be a very careful person or a very uncareful person or something like that. Again, it's conceivable, but if you don't have a concrete way of measuring it and then to work with it, you won't be able to really account for it. Sorry. So you need both criteria to hold. So you need it to be known and observed in order to be able to account for it. Okay, so what kind of multivariate analysis we can have? The first one is multivariate linear regression. So if you remember, we saw the example of a simple linear regression between two variables, y and x. So one way to write it down is using the following formula. So we can index it according to individuals or according to samples. So if we have, for example, we want to do some analysis over, I don't know, 100 samples, we can give each one of them an index, I. And then we say that the y value for the ith sample is some kind of constant number, which we call beta 0, plus some multiplication of the x variable for this I sample. And we multiply it by, again, a constant b, beta. And then we add some noise, which we typically call epsilon. And there are all kinds of different models. So sometimes people assume that those epsilons are normally distributed. Sometimes they don't. But anyway, that's you can also think about it as the residual, which we spoke about. So what's the actual difference between the prediction of the model to what you actually get? And like we saw, the null hypothesis under this simple model is that beta would be 0, meaning that guessing just a constant number, beta 0, for all samples is as good as making something that actually depends on x through this kind of linear estimation. Okay, does it make sense for all of you? Okay, so if we can say that reliably we see that a beta which is not 0 gives significantly better results, it means that y is dependent on x in some way. Okay, so having x gives us more information about y than not having it. That's the meaning of association, at least linear association in this case. Now we can then generalize the model to any number of variables. Very easily using this following extension. If now instead of 1x we have k different x's, x variables, then we can simply give each of them a different index. So now we have two indices. We have the first one indicating the index of the sample, and then the index of the variable. So we have k different x variables. And now we want y to be some linear combination of all of them together, again using some constant beta 0 and some noise epsilon. And each of them can have a different beta coefficient. Now, what we can then do is we can isolate a specific variable that we're interested about, let's say for the sake of simplicity, the first variable. And then we can obtain a p-value just for the simple null hypothesis that only beta 1 is 0. Okay, what it means is that the remaining k minus 1 variables explain y as good as all k of them. So if we accept this null hypothesis, it means that the first variable gives us no additional information on top of the rest with respect to the y variable that we're interested about. On the other hand, if we reject the null hypothesis, then it means that y is dependent on x1 after already accounting and controlling for all the other k minus 1 variables in a linear way. Okay, so that's a very nice way to actually account for these variables and see whether x1 adds additional information after accounting for them. Okay, if we see that we getting a coefficient which is not 0 gives significantly better results, it means that x1 actually adds more information on top of all the rest. But if we can set beta 1 to 0 and have sufficient information with the other coefficients, then it means it adds nothing more, at least under this kind of linear model.
So it doesn't make any assumptions on the other variables. The other variables either could matter or not matter. It doesn't matter.
Yeah. We're going to see some examples in which it could add troubles. I would say up front that generally it's not a good approach to just account for everything that you can possibly think about. And I'm going to illustrate specifically how it could go wrong in some extreme cases. Yeah, and all of those untested variables, which are not part of the null hypothesis, so in this case, x2 to xk, they're often referred to as covariates. Okay, so kind of variables that we add into the analysis, even though they're not like our primary interest. So we mostly care about y and x1 in this case. Okay, we only account for them. A nice extension that people sometimes use is called contrasts. So using contrast, we can have a multivariate linear regression that tests even more complex null hypotheses. For example, we can have some null hypothesis of the form that both beta 1 and beta 2 are 0. And then if we get a significant result, it means that either x1 or x2 or some combination of them adds additional information if we reject the null hypothesis after controlling for all the other k minus 2 variables. Okay, so you can actually test any kind of linear combination that you want, even something crazy. You can have some strange null hypothesis like beta 1 plus 1.7 beta 2 and beta 3 are 0 or any other linear combination. I mean, I don't think it's too useful to have those kinds, but you can have them. So you have the mathematical tools to formulate and to test those kinds of null hypotheses. But I'm not going to get into it today. OK, so that was linear regression. Now, a nice extension that we have is called logistic regression, which is used when y is binary. So if y is binary, linear regression is not so appropriate, because basically, like, the model allows any kind of continuous value. Okay, so if we have a y which is only 0 or 1, it wouldn't make any sense if our model produced something like 5. What does it mean? OK, so again, if we just want to test association with some variable x, Of course, with so many options, so we can use a T or U test or Fisher is exact, depending if X is continuous or binary. But again, we might want to test more than one variable or adjust for covariates. And then probably the best option we can use is the logistic regression model, which works very similarly to linear regression with a very slight modification. So in the logistic regression model, we have, instead of having y itself being modeled as some linear combination of the x variables, we instead take the probability of y being 1. So now what we get some linear combination, and then we use the sigma, which we'll explain in a bit what it does, but it makes sure that that the result will be in the range between 0 to 1. And once it's between 0 to 1, we can think about it as some kind of probability. And we think about it as the probability of y being 1. So if we get 1, we say that according to the model, we're quite certain that y is 1. If it's 0, we're quite certain that it's 0. Even if it's, for example, 0.6, then we think that there is like 60% of it being 1. OK, so what is this sigma function, which is sometimes called the logistic function or the sigmoid function? It has different names. It's what to see here. Like I said, it takes any number, so it can you can feed any number into it, and it makes sure that the result is between 0 to 1. And it has this nice property that it will get half, so it will be exactly halfway when you give it 0. So basically, if you give it something positive, it will give you something that is more likely tends toward 1. If you give it something negative, it will tend more 0. And it has this kind of asymptotic behavior, so it will tend towards 1 as x goes to infinity, and towards 0 as it tends towards minus infinity. And it actually converges very quickly. It's some kind of exponential convergence, and you can see the exact formula of this function here. That's what people use. OK, so to repeat, essentially the y variables determines whether to use a linear or logistic regression. OK, so if it's continuous, we use linear regression. If it's binary, we use logistic regression. And the x variables are not that important, so they can be either continuous or binary. So either way, we can fit it into the model. If we have something that is categorical, of course, we need to make it numeric in some way. So you cannot really put something categorical. And then you have a more appropriate and less appropriate ways to do it. So just choosing some random scale. So if you have something, I don't know, that gets 7 different values, it wouldn't be a good idea just to give them some random numbers like 0, 1, 2, 3, 4, 5, 6. That probably won't express the kind of nuances that you expect from this variable. More kind of a better way to do it is to use what people call one-hot encoding, and then you essentially break it to different variables. So you can have one variable saying whether it's the first category, a second variable saying whether it's the second category. Each of them is kind of a binary variable, and you can then take all of them together and put them into this regression model. OK. So yeah, we mentioned 2 ways. So we said a few words about stratification. And we mostly spoke about multivariate analysis. Just to give you a sense of when it's appropriate to use each of these approaches. So stratification has the advantage that it can highlight kind of associations that behave differently across different groups. So for example, you can see something that works only for males and not for females. And if you use multivariate analysis, you either get a significant or non-significant result, and you don't really see differences that could be important between groups. Another advantage that given the group, it's actually kind of model-free. So if you split it to males and females, you can then just run a simple statistical test, like Fisher is exact, or t-test, or whatever it is, within each of the group, and you make no further assumptions. Whereas if you use multivariate analysis, typically it will be some kind of linear kind of model, which assumes linearity, which could be appropriate or could be inappropriate. So that's like the advantages of stratification. On the other hand, you can have that the actual definition of groups could be arbitrary when you deal with continuous variables. So if you want to account, say, for the income of people, then it's not entirely clear like how you would split them into different groups. What are the thresholds and cutoffs that you use for different income groups? And very often it's underpowered compared to multivariate analysis, especially when the groups are not very large. So if you end up with very small groups, it's not likely that you're going to get significant results. And then it's a sign that you might want to use multivariate analysis. OK, so we're now going to illustrate how it works. We're going to see linear and logistic regression. And like I said, the per group analysis will be demonstrated in the upcoming bonus question.